It's almost time. Let's get it started. Hello, everyone. Welcome to the Single Cell Spatial Analysis Seminar. My name is Changyan Hu. I'm Assistant Professor in Cell and Developmental Biology, Michigan Neuroscience Institute, and the Single Cell Program. Today, we have our speaker, Dr. Michael Elowitz. Dr. Elowitz is a Howard Hughes Medical Institute investigator and Professor of Biology and Biological Engineering and Applied Physics at Caltech. Dr. Elowitz's laboratory has introduced synthetic biology approaches to build and understand genetic circuits in living cells and tissues. As a graduate student with Dr. Stanislas Liebler, Dr. Elowitz developed the repressilator, an artificial genetic clock that generates gene expression oscillations in individual E. coli cells. Since then, he has continued to design and build synthetic genetic circuits, bringing a build to understand approach to bacteria, yeast, and mammalian cells. He and his lab showed that gene expression is intrinsically stochastic or noisy, and revealed how noise functions to enable probabilistic differentiation, time-based regulation, and other functions. Currently, Dr. Elowitz's lab is bringing synthetic approaches to understand and program cell-cell communication, epigenetic memory, and cell fate control, and to provide foundations for future circuit-based therapeutics. His lab also co-develops the synthetic memoir system that allows cells to record their own lineage histories. Dr. Elowitz received his PhD in physics from Princeton University and did postdoctoral research at Rockefeller University. His honors include the HFSP Nakasone Award, MacArthur Fellowship, Presidential Early Career Award, Allen Distinguished Investigator Award, the American Academy of Arts and Sciences, and the election to the National Academy of Sciences. So please join me to welcome Dr. Elowitz.
Okay, thanks very much for that nice introduction. Let's see, I think, and thanks for coming, everybody. So I think what I thought I would try to do is tell you, let's see, mostly I want to focus on one idea that we've gotten very excited about lately, which has to do with a kind of circuit architecture inside cells that provides a versatile form of biochemical computation and shows up in a lot of different pathways and contexts. But before I do that, I'm just going to also want to tell you just about a couple of tools that we've developed recently, because I think, especially for this group, I'm hoping they might be useful. So with that, let me start. So the starting point for us really is always trying to think about the cell as a kind of little device, and an amazing device that can do all these amazing things, like differentiate into many different states, enable those cells in those states to communicate and coordinate with each other, form elaborate spatial patterns, develop into organisms, and generate things like the immune system that can patrol the organism and remove pathogens. And our basic understanding of how cells are able to do this incredible range of things has to do with a set of molecular and cellular circuits. And really the behaviors of cells and these amazing abilities come from properties of those circuits. And so over the last few years, or many years, we've been trying to kind of go through lots of different cellular functions and try to understand the relationship of underlying circuit architectures to those functions, whether it's communication, patterning, signal processing, and so forth. And at the same time, we've been also developing, and when we do this, we've been trying to kind of think about basic questions like, when you see a molecular circuit in the cell, what is it actually doing? What function does it provide? Why does it use one particular circuit design instead of another? And from a synthetic biology point of view, how can we start to program new functions in cells? And so those are kind of the big motivating questions, I think, for this sort of intersection between this field of systems biology and synthetic biology. And what I was going to say is that at the same time, we've also been developing tools that help us analyze and play with cells in different ways. And I'm going to talk a little bit about that first. So what I'll do is tell you about a couple of tools for recording and readout and delivery of RNA really briefly, and then talk about this theme of many-to-many protein interaction circuits. Okay, so one of the things that as single-cell biology kind of develops and spatial transcriptomics techniques have emerged, we have the ability now to look at tissues and and classify the states of individual cells directly in situ, which is really exciting. And one of the questions is, how did different cells get into different states? And part of the answer to that is usually there's some intrinsic cellular programs that are operating, and there's also extrinsic signals coming from other cells. And one of the crucial pieces of information that helps you discriminate intrinsic and extrinsic cues in cell fate control is lineage. So if you could look at a tissue, see the states of the cells directly in situ, but also reconstruct their lineage relationships. In other words, the lineage tree through which all these cells emerged, that could be very informative because correlations of cell state on the tree are indicative of cell autonomous programs, and lack of correlation is indicative of other extrinsic cues. Now, one strategy to do this is what's now called kind of genomic recording techniques, in which we try to have cells mutate barcodes in a controlled way, but stochastic way, and just put kind of random mutations so that if we could read out those mutations at the endpoint, ideally in situ, we could reconstruct this lineage tree in much the same way that we do phylogenetic inference from genome sequences of different species. So to do this, we really need three different kinds of capabilities. One is the ability to target stochastic edits to particular barcodes that have been engineered into the genome, the ability to read those out. And if we want to do it spatially, that means we have to do it through imaging, and then a kind of computational reconstruction approach. And so we've been working on this general idea for a number of years, but just recently, we've kind of, I think, got a system that scales in a much better way, and I think is really ready to be used to address biological problems. And the key, the core idea of this technology is to design barcodes that can be edited by CRISPR base editors, and they have some little tricks. So the modular unit of the system that I'll show you, which we call base memoir, is this sequence here that has an editable dinucleotide that can be targeted by a CRISPR base editor and go to any of three different endpoint states. So this AA can be edited by an A to G base editor to go to GA, GG, or AG. And critically, these three states are all terminal. So the AG does not then get edited to GG. And that's important because as editing occurs over time in these cells, we don't want everything to go to the same endpoint, okay? And so this is work from Kirsten Frieda that she initiated and has been really developed by Duncan Chadley in the lab. And by engineering sites like these and integrating many of them into the genomes of mouse embryonic stem cells, we now have cell lines that have about 792 different bits. In addition to these editable dinucleotides, there's also static barcodes that give you kind of identification labels for each of the edited sites, so that you can actually kind of identify which is which when you look at all these different bits that have been edited. So to show you what this looks like, if we have a mouse embryonic stem cell colony and we zoom in on a cell, then within the cell, we can read out by FISH the states of all of these different, the different mutation states by hybridizing probes that are specific for the unedited or each of the different target edit states. And then we also have probes that allow you to systematically read out these static barcodes that label each of the integrations. And so in this way, you can read out the state of all of these different bits and their addresses in individual cells. And so that gives you kind of the profile of what are the edits in each cell in situ. And that readout, I should say, is also compatible with using seqFISH or MERFISH to read out the state of the transcriptome by looking at the expression of different endogenous genes. So now we have cell state and we have these barcode edits in the same cell. And then the next challenge is reconstruction. And so we've kind of collaborated with Remco Buchart, who developed this Bayesian lineage reconstruction system called BEAST2, or originally BEAST, now BEAST2, which is used for phylogenetic inference in a variety of problems. And we've been applying it to reconstructing the lineage trees. And so this slightly fuzzy tree here is not because your eyesight is bad, it's because each of these little thin lines represents one possible reconstruction. And this Bayesian framework allows you to see kind of what are the most likely reconstructions given the data. And by doing this, we can now take a colony of cells and map out not just the states of those cells, which here 3 of the different states are shown in different colors, but also the lineage. And so here are the clades that correspond to some of the lineage relationships you see on the left. So this is a way to kind of see both state and lineage history at the same time. The other thing you can do is from this, you can also start to infer things about the dynamics with which cells have transitioned from one state to another. So this is an example of an inferred dynamic transition graph. It tells you the rates at which cells are switching between some of the different states we see in these embryonic stem cells. Okay, so this system I think now is kind of applicable in any system of interest that allows you to do cell engineering and spatial analysis. And we've also been building a different version of the system that's in mice so that it's kind of ready to go in any mouse, in a mouse that you could use to analyze lineage history or tissue of interest. And so if this is useful for people, please let me know. And also there's a paper describing it on bioRxiv right now. Okay, now that is great, but it's also true that it requires fixing the cells at the end to stain at the end point. And this is true of most techniques we have in biology, where even if they're single cell techniques, we're still killing cells to analyze them. And wouldn't it be great if we could analyze the dynamics of cells without killing them in a non-destructive way? And so for the last few years, Felix Horns, this really spectacular postdoc in the lab, has been working on trying to realize that vision. And the idea is to take barcodes again that are indicative of what cell you're looking at, but allow engineer cells so that they can package those RNA barcodes into extracellular vesicles, and such that we could recover the vesicles, sequence the barcodes, and then follow the dynamics of cell populations non-destructively. So to do this, he took a system called EPNs, engineered protein nanocages. These are self-assembling structures, proteins, de novo designed by Neil King and David Baker, published in, I think, 2016. And he outfitted them with an RNA binding protein called MCP that binds to this hairpin, the MS2 hairpin from the MS2 phage. And by incorporating into these engineered protein nanocages these RNA binding proteins, multimerized 8 times, we showed that they can very specifically and efficiently export just an RNA of interest. In fact, some of these systems are so specific that about 80% of the RNA that comes out in these secreted membrane-bound vesicles is the target protein. The other 20% is uniformly sampled from the rest of the transcriptome. And so what you get with this is the ability to kind of export a desired barcode in a very specific way. And what that enables you to do is then to do experiments where you can say, have cells, just allow them to export the barcodes over time and follow the dynamics of clones by just kind of taking time points, sampling the media without disturbing the cells, and then sequencing. And just to show you what this looks like, here's an experiment where we have a mixture of cells that are either puromycin resistant or zeomycin resistant, and we just collect supernatant at different time points. And so the regions between each of these little thin black lines is the abundance of one of these clones over time. This is just a small subset of the thousands of clones that we can actually track. And what you can see is that under puromycin selection, you get, as you would expect, proliferation of the puromycin resistant clones and loss of the zeomycin resistant clones over time. But the important thing is that we can see extinction events for each clone or growth of each clone by clone. And we can do the experiment and also select for zeosin instead and see the opposite behavior. So this just gives you a feeling for what you could do if you could recover these. And we're trying to move this system in vivo now. The other thing that's really great about the system is that if you add one more component to it, which is a fusogen, then these extracellular vesicles can dock onto target cells and deliver RNA. And that's important because mRNA technologies are really exciting ways to deliver genes and express proteins in cells, but delivering the mRNA is sometimes challenging. And so this opens up a paradigm in which you could engineer sender cells that could express or secrete extracellular vesicles containing an mRNA of interest, and then transmit that to other cells, which could express it to either kill the cell, change its fate, or perhaps produce a protein. So this is a paradigm for kind of RNA delivery from cell to cell, rather than using a lipid nanoparticle or something like that. And just to show you that this sort of thing can work, if we take the complete system and we try to export RNA encoding the Cre recombinase, we can activate a Cre reporter fluorescent protein in other cells here at a efficiency of about 50%, which is not too far below the maximum we could get if we just directly transfect RNA for Cre into the cells. And the other lanes here are just to show that this capability of cell-to-cell transmission of RNA requires all the different components of the system, the exporter itself, the fusogen, and the tag that recognizes the RNA. Okay, so we're kind of excited about this system. This is published last year. We call it Courier for controlled output and uptake of RNA for interrogation, expression, and regulation. And so we're kind of excited now to see, can we take this system and move it in vivo so that we can both track the dynamics of cell populations in an animal non-destructively, and also engineer cells that can deliver RNA to other cells within the animal. And again, if anybody's interested in this, please don't hesitate to let me know if we can help in any way. Okay, so now that's just sort of two tools that we've developed. I kind of wanted to switch gears and really spend the rest of the talk on one idea. And that idea has to do with this particular architecture that we see a lot and we've become very interested in. And I think it's an architecture, a circuit architecture that's connected to the advent of multicellularity. So if you think about it, long, long ago, there was kind of unicellular organisms that we can think of as generalists that had a single cell type. With multicellularity, we get specialization, many different cell types, each specialized in different functions, and they can cooperate with each other to give rise to more complex behaviors. Now, when we look at circuits in unicellular organisms like bacteria, we often see architectures that are involved strong and specific one-to-one interactions between proteins. I think the best example of this is in bacteria, the two-component signal transduction systems in which you have histidine sensor kinases, each of which interacts in a one-to-one way with one response regulator, which Mike Laub showed a number of years ago. But when we go to animal cells, like mammalian cells, for example, then we see architectures like this, where we have families of protein, variants, that interact in a many-to-many way with other protein families. So classic example are some of these core signaling pathways like bone morphogenetic protein, Wnt, Notch, FGF, and others, where you have each one of those pathways has a family of ligands and a family of receptors, and they're not one-to-one, they're many-to-many. We see at one level down where the different receptors can then interact in a many-to-many way with effector transcription factors. A good example of this is the GPCRs that interact with many different G proteins in a many-to-many way. We see it with the transcription factors themselves. They can dimerize or form higher order multimers, again, in a many-to-many way. So we have families of transcription factors like the BHLH family or the AP1 family that form lots of different dimers in a many-to-many way. We also see it with adhesion proteins, where, for example, protocoherins can interact with one another also in a many-to-many way. So this is a very common theme. And the question is sort of why? Why do we have this? Is it just an evolutionary artifact, or could it be providing some particular function? And a few years ago, we got interested in the idea that this kind of many-to-many architecture, superficially at least, resembles the sorts of many-to-many interactions you have between neurons and artificial neural networks. And of course, we know these are very amazing computational architectures. And is it possible that these sorts of biochemical many-to-many systems are also doing some kind of computation? Now, one thing that's sort of special about the the biochemical networks is that they are not just one network. There are many cell types, and each cell type expresses different combinations of these components. And so by expressing different combinations of the components, it's possible that each cell type is performing a different computation in this paradigm. So it's of interest not only to see how these systems behave as computers, but also how they vary from cell type to cell type in what they're computing. Okay, so what I'm going to try to do now is tell you 3 vignettes that are related to this theme. The first one is to ask how systems like this are deployed, which components are expressed in which cell types, taking advantage of single-cell RNA-seq atlas data. Then to ask how they perform different kinds of computation in the cell, and then to say how taking advantage of those computations, these systems could provide a basis for multicellularity itself by allowing circuits that can produce many different stable attractors. I hope that's where we're going. Okay, let's see how this goes. So the first part was work that was originally developed by Alejandro Granados when he was a postdoc in the lab, and also worked on by an amazing undergraduate here at Caltech who graduated recently, Niv Tanrar, and has recently been picked up by an also amazing postdoc, Judy Sho. And the idea was to think about when we have a pathway, suppose I have a family of receptors here. Could be like TGF-beta receptors, and there's really at least 12 different subunits you can think of for those receptors. But if I think about those, how they're expressed across different cell types. So imagine, this is just schematic, but imagine all the cell types in the organism, and I've grouped them here with this dendrogram according to overall transcriptome similarity. So maybe these cells are like different kind of T cells, and down here we have neurons that are very dissimilar to the T cells. And you can ask, how would I expect the pathway, just the receptor genes, their expression to vary with the overall variation in the rest of the genome? So one possibility is that cells that are more similar overall are also more similar in their, let's say, TGF-beta receptor expression profiles. I think that's a reasonable null hypothesis. And you might expect that there'd be reuse, so that, all of these T cells, for example, all used exactly the same profile of TGF-beta, but it's very different than what you had, let's say, in the neurons. In that case, we'd expect to see recurrence of the profiles, but it would be clustered on this dendrogram. A third possibility is that there is recurrence, the same profiles get reused, but it's uncorrelated with the overall transcriptome similarity. In other words, the same different cell types, even if they're very dissimilar from each other overall, use for a given pathway the same profiles again and again. And that's what I'm going to tell you we see evidence for. Okay, so the way to look at this is, here we took single cell atlas data from a variety of different projects, such as the Tabula Muris and Tabula Sapiens datasets, but others as well, integrated it into a single integrated atlas, in which each point in that in this data set represents a kind of cluster average transcriptome profile for a given cluster, global transcriptome cluster. And then we take that and we cluster it based only on the TGF-beta receptors, which you can see here are the rows. And so what you can see here is there's a lot of structure. So each of the thin vertical lines represents one of these cell types. And you can see that there's certain profiles of the TGF-beta receptors that recur again and again across these different cell types. The other thing you can see is that in general, the receptors are used in a very combinatorial way. Each subunit tends to be expressed in many of these clusters, and each cluster tends to express many overlapping patterns of different receptors. Okay, so if I make a UMAP projection of this integrated atlas in gray, and I just light up where each of these profiles occurs, you can see that most of the profiles are broadly distributed across very diverse sets of cell types. And what that's telling us is that it's not just that the profile of TGF-beta receptors is reused, it's reused in otherwise very dissimilar cell types. So one way to see this is, again, here in the columns, you have the different receptor profiles, different receptors. These are kind of some of these, the rows are the profiles, which we call motifs, if they are recurrent and dispersed across cell types. And what you can see here is just what these profiles actually look like on the left, and on the right, you can see how they recur across different organs. So what you can see is that most of these pathway, what we're calling pathway motifs, occur in multiple organs or tissues, and most organs and tissues have cell types that express multiple different motifs. Okay? It's very combinatorial. And that's a TGF-beta, what I showed you so far. But if we look at this across different pathways that we look at, we see similar kinds of behaviors with FGF and RAS and with the Notch pathway, with splice regulators, with a lot of different pathways seem to have this structure. So here's what it looks like for Notch, for example. Here's SRSF, the splice factors, and so on. Now, the other interesting thing is that if you look at which motif is used for one pathway and whether it correlates with the motif that might be used in another pathway across cell types, you find they're broadly not correlated. So each cell type might, cell types that might use the same motif for TGF-beta tend to use different motifs for Notch, for example, and vice versa. And so this gives us a kind of a sort of prefix menu view of cell type organization, in which most cell types are expressing the components from multiple pathways at the same time, but they're expressing them in different profiles, and they're sort of independent. So if you think of the appetizer as BMP, for example, different cell types will make different choices, whether they want the salad or the soup. The same Notch as the entree, you the steak or the pasta or the chicken, and so forth. So you can imagine kind of this mosaic organization of cell types, which I think is suggested by this analysis, again, of existing single cell atlas data. Okay, this also means that the pathway expression space is less complex than it could have been because there's a lot of reuse of these profiles. But it begs the question, which is, given that there are these special profiles that get reused again and again, what are they doing functionally for the cell? So does each of these profiles have a particular function? And to think about this, this is something we can try to address this question in the context of signaling systems in particular. So a number of years ago, we started looking at the bone morphogenetic protein pathway. It's a subset of TGF-beta. And we noticed that if you do two-dimensional titrations of pairs of ligands, you see different kinds of responses. So here on the x-axis is the concentration of BMP4 and BMP9. And you see that either BMP4 or BMP9 can activate the pathway. We're looking here at expression of a reporter gene. And the two combine kind of additively. So it's as if BMP4 and BMP9 are really doing the same thing. So even though they're two different variants. But we see different things with other ligands. So this is still BMP4 on the x-axis, but GDF5 on the y-axis. And now you see GDF5 can inhibit activation by BMP4 in a dose-dependent way. So the output of the pathway becomes a kind of computation of the ratio of BMP4 to GDF5. Or here with BMP4 and BMP10, you see either alone can activate, but they sort of neutralize each other's effects when they're combined with the wrong ratio. So we see a variety of these different computations in cells. And in a couple of papers, we were able to explain how those computations could emerge simply from competition between ligands to bind to different receptors. And just the competition to form different signaling complexes and the ability of different complexes to have different activities is sufficient to give you the sorts of computations that we observe. Now, the reason this is interesting is that it means that if we think about cell types that express different combinations of receptors, like those different receptor motifs that I told you about, they can then respond to different points in the concentration space of ligands. So for example, here, if I have the space of BMP2 and BMP9 for all of these heat maps, and each one of these heat maps is a cell line that has been perturbed for one or more of the receptors, then you see they respond to different points in this ligand space. So what you should think about is these different cells in an organism, different cell types, by expressing different combinations of receptors, even if they're exposed to the same ligands, some might respond and some might not. So for example, if you look at these points in ligand concentration space, different members of this set of cell types will be activated or not activated at these different points in ligand space. So this represents a kind of addressing system in which different ligand combinations can preferentially activate different cell types based on the receptor combinations they're expressing. Okay, so that was kind of what got us interested in this idea that there's a sort of computation going on through these many-to-many ligand receptor interactions. But one of the simplest, but also most important kinds of many-to-many systems that I already alluded to was transcription factors. So downstream of these receptors are basic helix loop helix transcription factors in the case of BMP, and they don't act as monomers, they act as dimers that kind of dimerize with one another in different ways. Another family that shows a lot of dimerization, homo and heterodimerization, are the nuclear receptors. So these are transcription factors that can homo and heterodimerize. So in this graph, I'm showing for each one, this self arrow represents homodimerization, An arrow between two components represents heterodimerization. And different cell types express these different nuclear receptors at different levels, as shown just as one example of these two cell types. So could these transcription factors, through these dimerization networks of transcription factors, could they also be doing some computation? And that's not a new idea. So my colleague, Barbara Wold, in 1993 had a paper where she essentially suggested the idea that thinking about helix loop helix factors, the idea that maybe what they could do, I'll just read you the quotes, is that they could act as these sort of flexible computational systems. So she proposed that dimerization among members of these helix loop helix family of transcription factors provides an attractive mechanism by which major changes in cellular phenotype could be initiated by altering only one or a few components. In other words, that the network of dimerization interactions could kind of respond in interesting ways to perturbations, and that these perturbations could kind of propagate through a network of dimerizing factors. Okay. And that the whole system is fundamentally combinatorial. Okay, so that's a kind of general idea, but we wanted to make it a little more concrete and really ask, how powerful are dimerization networks as computers? So what functions can they actually compute? To what extent can different cell types, by expressing different subsets of these components, compute different functions? And how hard is it to kind of fine tune or tune a dimerization network to act as a really useful and versatile computational system? Okay. So these questions are a little bit analogous to what's sometimes called expressivity in the neural network field. Like it's a term that means what kinds of computations can a neural network compute? And this is work that was led by an amazing student named Jacob, together with, as a collaboration with Ben, a fantastic postdoc, and a really wonderful collaboration with my colleague Andrew Stewart and his former student, Matthew Levine, who's now at the Broad. Okay. And the starting point was just to try to represent these networks mathematically. So we have a very simple model in which we say that any of these different transcription factors can homo or heterodimerize with the other factors with some affinity, so that the concentration of each dimer is just some Kij, some affinity for that particular dimer, times the concentration of the two monomers. This is just mass action. And we also impose conservation of mass, which is to say there's just a total amount of each monomer present in the cell. Then we can represent any particular network graphically. And here we have a scheme in which the size of this circle represents the total abundance of that protein in the cell. And the thickness of the arrow represents the affinity. So a thick arrow here represents a strong affinity here for homodimerization, whereas this heterodimeric interaction is very weak. And we assume, for example, here, no interaction at all. Okay, so with that kind of scheme, we can try to explore what kinds of computations are possible. And we can do it in two ways. We can build networks that have different affinities for dimerization among the different proteins. So here, the abundances are the same. The sizes of the circles are the same, but you can see that the pattern of arrows is different. Or we can take systems in which the pattern of arrows is the same, but the abundances are different. And that represents variation between different cell types. And so each of these possible ways of varying the network can give you, in principle, different kinds of computational functions. And if we look at the whole repertoire of different functions that are possible, we define that as the expressivity. And if we think about what functions are possible just by varying the abundances of the proteins in a single cell, we call that versatility. Okay, so just to show you how this works, this is really simple now. If I have just a really simple graph of two proteins and the output is a dimer, the homodimer of the second protein, the input is the first protein, you can see that adding the first protein will compete with dimerization of the second protein, and you'll get something where the input protein turns off the response here, turns off the dimerization of the output protein. If I add a third protein, I can go from an off switch to an on switch. If I then connect it, make it more completely connected, I can get a kind of bandpass filter with on and off combined. Or with 4 proteins, you can get kind of this notch filter. So you can do some simple computations with relatively simple networks. These also work in higher dimensions. So if I imagine there's two inputs to the system and one dimer is an output, I can get something like this that responds when I have one protein but not the other, or a NAND gate, which responds when I have, only when I have neither of the two inputs. With 5 proteins, we can get a system that responds in a kind of double bandpass, only to a particular window of concentrations of its two inputs. Or you can get more complicated things like XOR functions here that respond, when you have one or not, or one but not the other, or vice versa. So you can see that these networks, even simple networks, can do relatively complex computations. But what's really special about them is this property that they can do different functions. The same network can do different functions in different cell types. And the way to see this is by varying the concentrations of the other proteins that are not directly participating as inputs or outputs. And you can see in this example, as I just tune those proteins, I can change the point here at which this circuit responds to its inputs. Or here's another example in which, by a more complicated network, it starts as an AND, but by varying the concentrations of some of these other seemingly indirectly involved proteins, I can switch that to a NOR or back again. So this is the special property of these dimerization networks. They can do computations and they can change what computations they do depending on the levels of other proteins in the network. Now, one thing you might wonder is, well, really, how hard is it? Let me skip that. How hard is it to get a network that does something interesting? And so we got really interested in that question. Do these networks have to be fine-tuned to do exactly, so that all the affinities are just right, or, and it's very hard to find a solution, a network that will do the thing you want, or are there good solutions, good networks all over the place? So to do this on the computer, what we did is we just built random networks with random affinities, considered all the possible outputs, and then considered a repertoire of possible functions, and then tried to see if there existed a set of concentrations for the components that would allow that network to perform that function. And if it did, we said, okay, check mark, that we can do it, or not. If it didn't, then we said, okay, can't do it. And we kind of kept track of that. And the interesting result is shown here. So here on the x-axis, we have the size of the network, the number of different proteins. And on the y-axis, we have the fraction of all the possible functions that we know a network of that size could do that are done by that network. And each dot here represents one network. So what you can see is that there's a distribution of how capable each network is. But when you get to networks that are big enough, like 7 or 8 proteins, you start to see that almost all of the random networks, all these black dots, most of them can do most of the functions for some expression levels of their proteins. So what this is telling us, in a way, is that most random networks can do most of the functions. So it's like the random networks approach a kind of universality. And it means that it's very easy to find networks that are really quite capable from this point of view. So I think this is really interesting because the implication is that if we were to replay the tape of evolution in the Stephen Jay Gould sense, we would still get networks of this type, but they might have very different structure with different affinities. But statistically, they'd probably be very similar. Okay. So I also have to be careful. I don't want to say that these dimerization networks can do everything. They're, in a way, they're kind of the dumbest sort of network you can imagine because they don't have catalysis, they don't have ATP hydrolysis. The point is that even though they're relatively simple, they can do a lot of functions, but there's still things they cannot do. They can't do magnitude amplification and things like that. Okay, so for the last little vignette here, I want to take it one step further. So far I've been talking about networks that kind of process information in a feed-forward way. So signals come in to ligands, come into receptors. That controls the activity of the pathway. That gets processed by some transcription factors and activates target genes. But real circuits have feedback. And that's important when you think about the problem in multicellularity of how do we go from some pluripotent stem cell into a huge array of different cell types, any of which can be stable. What are the network architectures that generate specific cell types and constrain the transitions that you can get from one cell type to another. These circuits have to have a lot of interesting properties. They have to allow kind of progressive commitment, as you see illustrated in this diagram of the hematopoietic system. They have to allow steering from a stem cell into lots of different cell types using the same signaling pathways like Wnt and FGF and TGF-beta and Hedgehog again and again. And they have to have the property of being able to expand during evolution, because we know that a lot of the same transcription factors that generate and specify cell states in simple organisms are still there in higher organisms, but they've just duplicated and the networks have become more complex. But it seems like over evolutionary time, these networks have an evolvability property that allows them to expand. Okay, so one of the frameworks for thinking about how networks how you could get different cell states that could all be stable goes back at least to Max Delbruck. It was the idea of bistability or multi-stability more generally. And this is a little sketch that he had of two metabolic pathways that were cross-inhibiting each other. And he pointed out that this sort of model could account for large numbers of alternative steady states similar to the phenomena of differentiation. The point is that you could get a situation in which one would be active or the other, but not both. They could both be stable. So from a mathematical point of view, we can think of this kind of system this way as a sort of dynamical system in which the behavior of the cell depends on the concentrations of two or probably many transcription factors. And we can think of systems where there's these stable attractors, these are these black dots, such that if the cell starts at one of these states and is perturbed a little bit, it'll go back. But if you perturb it enough, for example, by overexpressing a transcription factor like MyoD, you could cross a threshold and then the dynamics would drive you to the other fate. So if this is like a muscle fate and this is a fibroblast fate, that could kind of, just in a qualitative way, explain this phenomena of reprogramming. But this is a very appealing, very simple conceptual picture, but we don't really know if these kinds of dynamics could work in real cells with all of the stochasticity, noise, unpredictability, and complexity that exists in a real cell. And so a few years ago, we kind of wanted to know, is it possible to engineer a synthetic cell fate control system from scratch? And my former student, Ron Zhu, took on this project. And he tried to think of what kind of circuit design would do this. And the idea was to kind of look at the features you see in natural circuits and see if we could be inspired by those. And a lot of natural cell fate control circuits, as I've already told you, are based on transcription factor families that undergo homo and heterodimerization. So for example, the master transcription factor for myogenesis, one of them is MyoD, and it can heterodimerize with E47 and activate a bunch of myogenic genes, but it can also heterodimerize with other factors like the ID factors to produce inactive complexes that do nothing. And so these are sort of two themes you see in natural circuits, autoregulation and dimerization. And so with Ron, we designed this circuit we call multi-fate, which is the simplest circuit design that incorporates both of those features. And briefly, the way it works is you have, imagine that for the simplest version of this, I have two genes, A and B. A can homodimerize, and the homodimer, but not the monomer, can activate more A. That's autoregulation. Same thing for B. But they can also, in analogy with the MyoD ID complex, they can also form inactive complexes that do nothing, that are just inactive heterodimers. So we can describe this system mathematically. And when you do that, you can find regimes in which you can get things like tri-stability. So here's a regime where you have like three black dots. Those are attractors. And any of those three is stable. There's many other regimes as well that I won't go into. Okay, so Ron engineered proteins that could implement this circuit based on zinc fingers. And he was able to engineer them so that they could activate when they're homodimers by adding homodimerization domains, but not when they're just monomers. And he could further mutate them to kind of really get rid of all the residual activity of the monomers while preserving activity of the dimers. So these were the components we needed to control the system. And we also engineered other little control knobs onto these things like inducible degrons, so we could control protein stability, the ability to control dimerization with the drug, other inducers, and fluorescent protein reporters as well. So using those components, you can build this multi-phase circuit with two genes with two different zinc fingers integrated into CHO-K1 cells, and then activate the circuit by adding the drugs that allow dimerization and protein stability. And you get different behaviors in different clones depending on the exact number and sites of integration of the components. But for some of them, we see things like this, where we turn on the circuit and cells go to either of these three states. And that's somewhat analogous to what you see in the model with tri-stability. Critically, these states are stable. So if you fax them out, cell sort them out, and then culture them for long periods of time, each cells in one state remain in that state over long periods of time. I should also say, I'm not going to show it, but they're not stuck in those states epigenetically, so we can still switch them from state to state by perturbing the transcription factors. Now, the thing we really like about this design is that it's expandable. So if you plug in a third transcription factor without re-engineering the existing two transcription factor circuit, that third one can participate and form other inactive complexes with the first two. And this can generate phased behaviors like this, where now instead of 3 states, we have 7 stable attractors in the space of these 3 transcription factors. And so this can be built, put into cells. And if you look at those cells, so here, what's nice is that each of the 3 proteins has one fluorescent protein, either red, green, or blue. And so different states correspond to different combinations of those 3 colors. And single cells sorted to be in one of those 7 states remain in those states as they grow into colonies. So this is just a nice visual way to see that each of the 7 states is stable by itself. Okay. And the stability can go for a long time. Now, the systems are expandable beyond 3. So more recently, my student Evan said, why don't we add a 4th transcription factor to get even more states? And now he's shown that he can get 15 stable states. So if you sort cells from any of these 15 states, not the state where all proteins are off, but every other state, they're still in those states 12 days later. Okay, I think I'm going to skip this. But these systems have other interesting properties as well. Okay, so how far can this go? Well, if you, in the model, just imagine increasing the size of the multi-phase system by adding more and more transcription factors that participate in this dimerization network, you get exponential growth of the number of states up to a point. Eventually that saturates, and it saturates for an interesting reason, which is that as you have more factors, the number of heterodimers grows as the square of the number of factors, whereas the number of active homodimers grows only linearly. And so eventually you just have too many different inactive species. But nevertheless, based on the parameters we see in the experiments, we think that these systems with a handful of transcription factors could support hundreds of different cell states. Okay, so just to conclude this section, I told you is that this multi-phase design is naturally inspired. It just has 2 features, combinatorial dimerization, autoregulation. But with that, it can produce these different properties, multi-stability, the ability to switch states, irreversible transitions I did not show you, and progressive differentiation, I also didn't show. It's expandable. It agrees in it to a striking extent with the models for this circuit. And we're thinking of it now as a basis for engineering synthetic multicellularity, division of labor, and other sorts of behaviors like that. It's also not totally omnipotent. There's lots of limitations with it as well. Okay, so just to summarize the whole talk, these many-to-many interactions are really prevalent in multicellular systems. And when we use single-cell RNA-seq atlases, we can see how, some of our favorite pathways are deployed in a very interesting way in terms of combinatorial motifs that get reused in different cell types. These motifs probably correspond to different functional behaviors that process information from different incoming ligands or incoming receptors, and then use that to control different downstream genes. So we think of this now as kind of layers of many-to-many networks that process information at many different levels, and combined with feedback can even give you properties like multi-stability. Okay, let me stop there and just I tried to acknowledge a lot of people along the way, but this work is really the result of an amazing group of very talented people who are in the lab or recently were in the lab who did a lot of this work. So thanks very much.
Well, thank you so much, Dr. Elowitz, for the wonderful talk. So everyone, please feel free to mute yourself. We have 10 minutes of Q&A. Let me get started with a quick question. So how much role do you think the time axis plays in the computation of many-to-many networks? For example, the binding duration or, yeah, thank you.
Yeah, that's a great question. So what I focused on here is a pretty static analysis where we look at the steady state behavior. But these systems can also be dynamic. And in fact, natural dimerization networks we know are dynamic. The Hess-Hay system, these basically the helix factors are parts of oscillators and some mitogenesis, for example. And so the way we think about it is there's a separation of timescales in which the many-to-many interactions are fast because they're protein interactions, and transcriptional regulation is slower. And so we think they can be part of more complex dynamic networks, broadly speaking. Yeah. Sorry, that's a long-winded answer, but yeah, I think that's a really exciting question.
Okay, thank you. Very exciting.
Hi, I have a question. So, beautiful talk. So, in our body, about 200 different kinds of cells we have. Then, in your theory, so how many transcription factors are sufficient to make our different cell types available in our body? And the follow-up question is, we usually have many terminally differentiated cells, and the proliferation and differentiation appear to be coupled. So is there any model where those proliferation over terminal differentiation it can be explained in your model?
Okay, so I guess the first question is sort of your first question has to do with, we have a certain number of cell types, hundreds, maybe thousands, probably, of different cell states in our bodies that are, and this system can produce, okay, the multi-phase system in principle could scale to produce hundreds of cell states. That's what I showed with a handful of transcription factors. Now, we have hundreds of transcription factors and probably a larger number of states. So I don't think that multi-phase, exactly as we've done it, is a good model for to explain all of the different cell states in the body. Probably there's what it does is it kind of abstracts out a particular aspect of these cell fate control networks, but the real networks are more complicated. And there's clearly more factors, and different subsets of factors are working in different cell states. So I think what the value of it is not so much as a literal model of exactly what's going on, but more just showing from a minimal point of view, what are the minimal requirements to get multi-stability? So that's the first question. I think your second question was about the relationship of what terminal cell fates to
terminal kind of
proliferation
profile versus kind of proliferation. So usually those stem cell populations are proliferating cells, and once they become terminally differentiated, or kind of stopped proliferating, kind of their cell type is almost like a fixed. So is there any model to explain what's happening there?
Yeah, so that's a good. So I think if we take multi-phase just as literally, then if we turn off proliferation, we reduce dilution, we get rid of dilution of the transcription factors. And so within multi-phase, it is easy to see how that could make the system more stable. But I don't want to overstate that, because I think in real systems, we have also chromatin level, epigenetic regulation, other mechanisms as well. So I guess the short answer would be, I think there's a way in which proliferation could influence the behavior of the circuit and make it more stable, or sorry, lack of proliferation could make it more stable. But I don't want to overstate that and say that that's the whole explanation.
So we have a question in the chat window from Tom. Many biological networks are tolerant to the loss of nodes. So what type of synthetic networks exhibit the tolerance of one or multiple nodes or components, which ones don't?
Oh, okay. Yeah. So one explanation for why we have this seeming redundancy of very similar factors that do similar things is that maybe it's just a kind of fault tolerant mechanism so that the whole system is robust to loss of a component. And I guess you're asking what kinds of systems exhibit that property. Yeah, I mean, I guess in a way, there's sort of two viewpoints here. One is that there's true redundancy, and the redundancy gives you allows you to tolerate loss of effect factor. Another view is that what appears to be redundancy is the fact that sometimes, and this happens, for example, with TGF-beta family ligands, where you can delete, one ligand might replace, or one receptor, this has been shown, can replace a different receptor subunit in most contexts, but you'll have problems in some other context. So it's possible the replacements are partial, that they're partially redundant, but not fully redundant. And I think that's more consistent with a view in which the many-to-many interactions are there, kind of process information. And even when two components look like they're redundant in one context, they may not be redundant in another context. If that makes sense. But I don't think I have a general answer to your question, actually, but I think that's the way we're thinking about it.
I also have one question. So at the atomic and molecular scale, the interactions of these molecules sometimes are influenced by fundamental laws in quantum mechanics. So do you think the many-to-many networks might end up being a quantum network that have some features that is very different from, like, for example, artificial neural networks?
Wait, sorry, can you explain what you mean by the quantum network? You mean like quantum mechanical?
Yes. Yeah, that is not the classical, for example, the networks that we know in the artificial neural network, but there might be some interesting quantum features in the network. Yeah.
Yeah, I don't think nothing in the way we've approached this has any quantum mechanical real description. It's just mass action interactions of protein binding. But so I haven't thought about that really.
Got it. Got it. We have another question in the chat from Jianping Fu. So in the multicellular context, spatial information becomes important. The extracellular transport and morphogenetic genetic features. Do you think if it is possible to incorporate these features?
Yeah, this is really important when we think about these signaling pathways, because it's not just how much protein is there, but also how much is it at the surface and how accessible is it at the surface. I think it is possible to incorporate those features, but it requires protein level measurements at the level of single cells in the context of real tissues. And that's something I think needs to be incorporated in this framework as well.
Yeah, maybe some of the real-time optical fluorescent reporters could help to collect some of the data to address this question.
Yeah.
Can I ask one question? Yeah, so thank you so much. This is like a fantastic talk. And also I attended at the middle of the talk, so I missed some. But you study cellular states and engineering. Have you ever sought any evidence like the cellular states that does not exist in our body? So can you synthesize a new type of cellular states by augmenting the capacity of the transcription factor, for example, and then that may get useful for some application purposes? But just curious about that.
Yeah, I love this question. It's does the cell fate control system that we have in our own cells produce states or tractors that exist and could be accessed but never appear maybe in normal physiology, but are really there? They're kind of like ghost states or something. And I think people have seen evidence for such states. For example, when they try to reprogram differentiated cells, I think Catherine Plath did this, and I think other people have evidence for that those kinds of states could exist. And a fundamental question that I don't know the answer to is whether any system that is sufficiently complex, in other words, can produce a sufficiently large number of states that you want, does it inevitably require that it also have the complexity to produce states that you may never use? And I think, yeah, I love that question. I don't know the answer to that.
But thank you so much. These are exciting to see. Yeah, thank you so much.
Wonderful. So it's almost time. I'd like to thank again our wonderful speaker, Dr. Michael Elowitz, and thank everyone for joining us, and have a wonderful day. Bye.
Thanks very much. Really enjoyed it. Thank you.